Dear ladies and gentlemen, dear students, dear professors, first of all, I would like to thank the Szeged Scientists Academy for supporting me and giving me this prestigious award. And then I would like to talk about the development of a novel drug delivery system for the brain. And it is important because the problem is that many neurological diseases, like Alzheimer's disease, Parkinson's disease, epilepsy, or brain tumors, are especially challenging to treat pharmaceutically because the majority of drugs cannot cross the blood-brain barrier, which I'm sure that you've heard about today in the afternoon in Professor Maria Deli's lecture. So as a solution to that, in our lab, we take the compounds to be delivered to the brain, we load them into nanoparticles, and then specifically target these nanoparticles to the blood-brain barrier. And one promising new group of candidates for this targeting process are nutrient transporters present in brain endothelial cells. They are available in large numbers, and they have a high transport capacity, yet they are very much under-investigated. So our hypothesis was that if we decorate the surface of our nanoparticles with the ligands of these transporters, or if we use the combination of different targeting molecules as targeting on the surface of nanoparticles, it will result in a better permeability across the blood-brain barrier. So for that, we made 4 types of nanoparticles from non-ionic surfactants and cholesterol, therefore they are called niasomes. We made an untargeted one targeted with the amino acid alanine, one with the tripeptide glutathione, and one with the combination of the 2 molecules. And we chose a high molecular weight fluorescent protein called Evans blue albumin complex that is known not to cross the BBB as a cargo of these nanoparticles. So as you can see here, the diameter or the size of these nanoparticles were around 100 nanometer. They had a low polydispersity index and a slightly negative surface charge. So first we investigated how well do the cells, how well do brain endothelial cells take up the cargo of our nanoparticles. And for that, we treated the cells with the solution of nanoparticles. And after 4 hours of incubation, we washed the cells, lysed the cells, and quantified the fluorescence intensity of our cargo molecule by fluorescence spectrophotometry. And as you can see here, the unencapsulated or the free cargo molecule has a very low entry to the cells compared to the non-targeted niasome control, whereas the presence of our targeting molecules on the surface of niasomes in all cases elevated the cellular uptake process, double targeting being the most effective, producing approximately 2.5-fold increase in cellular uptake. We could also visualize this by confocal microscopy. Here you can see the nuclei of the cells in blue and the fluorescent cargo of the nanoparticles in red. Next, we investigated how well do they penetrate the blood-brain barrier, and for that, we used our lab's signature co-culture model, where we culture together brain endothelial cells, pericytes, and astrocytes in a transwell system where an upper compartment or blood compartment and a lower compartment or brain compartment are being formed. And we treated the upper compartment with nanoparticles for 4 hours, then quantified the fluorescence intensity of the cargo from the lower compartments. And you can see that the trend is very similar to the uptake experiments. We had a very low penetration of free or encapsulated cargo molecule, whereas the targeted nanoparticles penetrated the blood-brain barrier better. So as a summary of this first part of my presentation, we prepared vesicular nanoparticles from non-ionic surfactants, and we showed that both the uptake and the permeability of our cargo was higher when we decorated with targeting molecules, and that double targeting was even more effective. So therefore, we concluded that we could deliver drugs across the BBB with this combination. But here comes the question, then what? Because in many cases, neurons are the therapeutic targets. But to get to neurons, these nanoparticles have to travel through layers of brain endothelial cells, pericytes, and astrocytes. So our aims for this project was to verify the gene expression pattern of relevant transporters and then test these cells for cellular uptake, and also we would like to know the mechanism of cellular uptake. First, with the help of Dr. András Tóth from the Department of Biotechnology at the University of Szeged, we verified the gene expression patterns of proteins that transport alanine into the brain in these cell types, and you can see the data here. Then we performed uptake experiments the similar way as we did before, but we also investigated the mechanism of uptake by introducing some inhibitory conditions like 4 degrees sodium azide, philippine, and cytokalasin D. So at 4 degrees, all the active processes in cells stop. And sodium azide is also an inhibitor of ATP production, whereas philippine and cytokalasin D are inhibitors of different endocytic routes. Philippine is specific for caveoli-independent endocytosis, whereas cytokalasin D is more of a general inhibitor. So you can see here first in primary rat parasites that the double targeting or the double targeted niosome resulted in a 2-fold higher uptake, and that in all cases the uptake process was or could be decreased with the treatment at 4 degrees or with sodium azide, therefore suggesting an active energy-dependent process. And we could also decrease the level of uptake by these endocytic inhibitors, philippine and cytokalasin D. The tendency is very similar in primary rat astrocytes, but the extent is different. So we could only produce around 1.25% increase of cellular uptake. And you can also see that the inhibitions were quite similar. And we also tested it in human neurons, which you can see that it again produced a 2.1-fold increase in the level of uptake and a very high inhibition of endocytosis. So as a summary of the second part, we concluded that the level of uptake of our double-targeted nanoparticles were significantly higher in parasites, astrocytes, endothelial cells, and neurons too. And we also investigated the mechanism, and we can say that this is an active, energy-dependent process. It is partly by endocytosis, mediated by endocytosis, and it happens on different endocytic routes. So as a conclusion, we say that alanine glutathione double targeting of nanoparticles could possibly deliver cargo to all the cell types of the neurovascular unit. So as future plans, we plan to image the cargo in living cells, which is ongoing right now. Here you can see a picture of that. But we would like to also monitor the intracellular fate of nanoparticles by co-localization studies, and also we would like to encapsulate certain kinds of drugs into the nanoparticles, for example, anti-inflammatory drugs. So with that, I would like to thank my mentors, Professor Mária Deli and Dr. Silvia Veselka, for their support. I would also like to thank Dr. András Tóth and Dr. Rákai Gábor from the University of Szeged. I would also like to thank the Szeged Scientists Academy. And finally, thank you for your attention.